So welcome to the CRI. Welcome everyone, and good to see some faces I know, and even more exciting to see faces that I don't know. That's the whole idea of the CRI, to put people together and co-construct and do something useful with the brains, the expertise, the different disciplines, and different ideas of people. I changed already 3 times in the last 20 minutes the slides and the things that I wanted to share with you. So I thank Alex and James for a brilliant presentation of both what is synthetic biology, what can be done with synthetic biology. What I'll try to do is tell you a few words about what we're doing here, why I think it's relevant to have such workshops here, and few ideas of where we take synthetic biology from here. But mainly the main thing about the CRI is that what we try to have here is a culture of questioning. Okay? So you heard 2 talks that probably at least 50% of the things that were said were not understandable, and probably have other questions. So before I go further, any questions, anything that was not understood, anything you wish to learn more that I can help with, and that's the time for you to talk finally. So wake up, you can raise your hands, go, come on. Spread yourselves a bit, stretch yourself. What of the things you heard until now merits a bit more focused explanation?
Maybe we can open the window and it will help.
Sorry?
We can open the window.
We can open the window. Yeah. 1 minute of air.
Thank you very much. Maybe I could start it up. Thank you very much for your inspiring and popularizing presentation. I'm very sorry for the delay of the question.
If you could speak louder, please, Karim.
But I'm shy guy.
Okay.
Okay, so thank you very much for your presentations, both of you. It was quite a lot of stuff to recall, try to understand what I cannot understand. Any questions? From an industrial point of view, I'll be representing the group set, the domestic appliances company here. For both of you, what is for you the timeline, the realistic timeline of what is going on, what we will be doing, what will you and other people from the design part and the scientific engineering part will be doing by tomorrow, and what is the part of the dream in what you are doing and what you have shared with us. Usually when you are very passionate about something, it's very easy to, and it's very good to share that passion with people, but when they're beginner, just like I am, it's really hard for me to be able to understand where are the 80% and where are the 20%. Where's the reality, 20 or 80?
Maybe I'll try to start answering, and then James and Alexander can join in. Okay? I'll be very blunt. Okay? Anything that you design today that will stay in closed doors, okay, meaning that in a certified lab conditions for using genetically modified organisms, you can make your product tomorrow. So if the product you're interested in is a product of a metabolic pathway of the living system, but it's not the living system itself that you put to the use of people, tomorrow you can do it. Everything else depends on a very delicate, I would say, equilibrium, right, between risks that we do not know how to identify yet, right, and the positive effects or the positive impact of your invention of what you want. Basically saying, you know, if you come up with something that would save lives that no other technology would do, right, then it's more likely to happen in the next 5-10 years. Anything else, we don't know yet. I don't know if you want to make your own more optimistic way of looking at it, maybe, but
I think it very much depends on what you're trying to do. I think for me it was very interesting to see that some things that you imagine are easy and some things you imagine are difficult are actually the other way around. So engineering a bacteria to do something is quite easy. You could do it today with very, very simple equipment. We could do it with a bucket of ice, a hot water bath, and a few chemicals, and we could make a bacteria glow under fluorescent light, and that bacteria does not do that naturally. So some of the processes can be done by anyone, and that makes it possible for everyone to get involved if the regulations and if society allows it. However, much more complex things, so making a bacteria that can look for different chemical signals and change colour is very, very tricky to do. So this arsenic biosensor that I showed, the one that will detect pollutants in water, that work started in 2008, 2009, and it's still happening now. We're still working through the process of developing it. And that's just one very simple sense arsenic of change colour. Like, very, very simple idea conceptually. But to actually engineer it effectively just takes a very, very long time to do. So I would say that the design is difficult. It's the engineering that's very difficult to do. But the actual manipulation and transformation is very, very easy to do by comparison. So I'd say that some things are very far away, some things are very close. You have to look at the specific.
So if I sum up, you've got two different answers. One saying there's a process, and the process can take a long time. And the other is, even if the design is perfect, what would make this an actual product that would be in the market? I think that basically we got two scales.
Just use the mic, please.
Other questions?
Just the mic, please. Can you just use it because we are
Other
recording.
questions, please.
Yeah, it's not for the song. It's just we are recording. So yeah, thank you so much.
Yeah, just to elaborate.
So maybe one before you start. Sorry, I'll be rude. Okay? There is one rule in the CRI, okay? And this is students get to ask questions first. Okay? And this is a very simple reason, because if all the experts here will start asking questions, right, there will be even more inhibition for the students to ask questions, right? And for those who didn't bother to read the slide all the way to the end, the answer is actually there. So, students first, questions.
Yeah.
Matt,
Should I just yell?
Yeah.
So I have a question for designers, and that's how do you handle what you don't know in your design? So for example, the scale of what's alive and what's dead. So a lot of that, making that undead cell, we don't know what steps you'll need to do in what order to get it to work. So when you're designing something like that, how do you know how do you design something when you don't know what it is or how it fits together or what the pieces even are?
I think probably Alexandra should answer this as well. But I think it's important to, when you're a designer, to not let your ignorance hold you back too much. Obviously, you should, if you know something is wrong or false, you shouldn't suggest otherwise. But if you don't know what is real and what is not, what is possible, what is not, then I think it's okay to make a proposal that may be in that grey area somewhere, because it helps people to ask questions and answer questions. So I think if I'm ignorant about something, I don't worry about it too much. I try and get as much information as I can, and I just work until someone tells me, no, that's wrong, you can't do that.
I quite agree with you. It's basically hypothesis-driven research. Even if you don't know how it works, you think of a hypothesis, how it might work according to you, and then you try. If there is experimental evidence that shows that your hypothesis might be right, then you go a step further. But if there is evidence that shows that what you think could be the way to evolve is not the right way, then you have to think of another hypothesis and try again.
And I'll add as well. So my point of view as a researcher, and this is I think where you can make a distinction between an engineer and a researcher. So I go back to Feynman's famous quote that James already put there.
Stole my slide.
Sorry?
Stole my slide.
I stole your slide. That's the original words that he left on the board in one of his last lectures in Canada. But basically what I'm trying to say about this very question is that as a researcher, when I try to reconstruct a system, taking an engineering point of view, nothing gives me more pleasure than seeing that it fails. Why? Because what we're trying is to understand a living system. Right? We try to get an approximation of it. If we're all right and we know everything, I'm out of job. Okay? I have to do some other profession. And I think it's exactly where the delta, where a system doesn't work and you start making it work, right? You start understanding. This is the beauty of this kind of engineering synthetic biology approach. So I hope this answers your question. I'll take one or two more questions and then I'll share some more stuff with you. Yes?
So the question about for James and what do you think about the legitimacy of a designer between creating a real functional product and creating a fiction about the biological system? How do you manage that? Do you have some preferences, or do you think the designer should be more implicated in one of these?
Okay, so I'll go first. So I think that both are great, because this is basically the great thing about designers is that you think the other like completely differently than scientists, and what for scientists might seem like an impossible project, and he would never, or she would never think about it. A designer might have an idea, and the scientist might think, oh yeah, but maybe it's not impossible. We could do something that seemed impossible, actually a real product. And also, on the other hand, for producing something real, the designers also have a great bonus of helping the scientists also think of some actual products.
I think that all design is about imagining some kind of future. So I think even if you're designing something you intend to make tomorrow versus make in 10 years, I think it's still the same fundamental process. Having said that, I think it goes back to what we talk about when we think about what's possible and what's not possible. So I think it's very important if you're speculating to try and understand as much as possible. So to try and get rid of your ignorance as much as possible and know what the most senior expert knows in order to build on and speculate on top of their understanding. So again, for me, when I was designing the meat, I thought that meat would be very easy and that maybe in 5 or 10 years we'd be seeing this thing in shops, but it still costs $100,000 and you still have to use very nasty products to make it. I'll tell you more about this later. But the genetic engineering, which again to me I felt was much further away, is actually much closer. So it's really about getting into the lab and understanding it from the scientist's perspective, what's difficult, what's not, what are they doing now, what problems are they struggling with, and then from that point you can have license to speculate, to design something that you're not sure whether it's possible or not to ever achieve it.
Am I understanding your initial answers right. Like if I understand it like that, which is usually the science behind all this is fairly straightforward, and what really takes time is scaling it or making it risk-free or risk acceptable.
No, I think it's every project, every research question has its life of its own, and I think James pointed out very nicely. Sometimes things work extremely well, very fast, and sometimes things that you think that would take years to develop actually take years to develop. And what I do not know how to create, I do not understand. It's beyond this is the fact that there's much that we do not understand. And so, yes, in both scales it can be stretched. Artemis.
It was just to add something on top of this, because we discussed a lot about rational design and straightforward design, like I know what I want to do. I say, okay, what kind of parts do I need to do this, how I implement this, how I have this working great. But what is also a reality in synthetic biology and in the industry that is behind it is what we call directed evolution. And so it's another also way that is used in complement with rational design, and where instead of really saying I will use this gene and this protein, you're more creating an environment where the bug, a bacteria that does what you want, will be selected and will have offspring, and you will generate new mutations. So nature finds its own way towards the solution. So you can also think of how do you design an environment that helps you produce what you want. So it's also something that designers should think of, because it's like, in my opinion, creating a space or a lab, or it's hard to have people sharing things. You have to create an environment, and it's hard to do this for bugs, and I think it's also some way where they are similar.
Almost a planted comment, but this leads me to what's the CRI all about, right? Because the CRI is all about making such an environment for people that can combine their expertise and come out with something that they never thought that would come out of this. So, very briefly, some sort of principles behind what we do here, and there is only one good correlation that we know between life expectancy, right? It is how much time you're going to spend in this world, right? And it's not the color of your hair, and it's not the age of your parents, but it's overall the level of education. Okay, so you can see sort of literacy percentage in different countries and life expectancies in different countries, and that's real correlation. Okay? So treating health questions and treating education questions is one thing, and what links these two is research about education, about how we learn, and research that has to do with better health, namely life sciences. And this is sort of the core of what happens in the center. And I will not go over all the different programs that, sorry, I'm not going to the wrong direction, I guess. I won't go over all the different activities we have here. It's undergrad, master, PhD programs, and lots of citizen science, computational biology, and systems biology approaches to different questions in real lab settings. And I want to focus on one or two subjects that we have here. And so this is the build-up of the CRI. I want to focus on recent years. First, the CRI has a new space. So this is one space that has to do with the biology lab, and that is an open lab, and you will see it in a few moments. And we have a new space in the Marais in Charles-V. You're all invited to come to see it. And within 2 years, the labs will move there. And one of the things that is already open there is an open lab. Artemis that just talked there, if you want to learn more, he's the person to talk to. The idea is to put together people that come from design, from education, from researchers, that come up with prototypes of how to do better research, education, etc. And all this with open technology, frugal science, so it will be available for everyone. Okay? So inclusiveness in the actions that we do is another key point. And another thing that I would like to point out in our new activities, and that's brand new, it's a new master program track that is directed by Sophie Penn, and some of the designers here at least should know. And it has to do with the same spirit of the Open Lab, put together designers, researchers, people that come from education, teachers, and design both the environment, the content, the tools to teach and learn through research. So anyone who's interested in this, shout out, talk to Artemis that is here with you during these days, or you can write directly to Sophie. And I think that's sort of the crux of it all, right? And that's why I try to limit what I'm saying. People try to look at the retention rate, okay? How much do you retain of what I'm saying today? And that's probably 5%. Okay? But if you start learning by discussing, which you do today, or by working on a project, or teaching others what you think you know, okay? This is where you really start learning efficiently. So this is what we're trying to do here at the CRI. So this is to summarize wake-up call from what you heard already from James and Alex. And what we've been doing here, like James showed his work with the e-chromite, and we take every year a group of students, and some of them never did synthetic biology before. Those that already participated in this adventure, there's 1, 2, 3, 4, 5 people already, so you can hear more. And what you see, they come up with their ideas, and the ideas are very, very different, very, very different from each other. They try to think of how to make bacteria that are individuals, okay, as a community, as a multicellular organism. That was back in 2007. They tried to have a clock made out of bacteria. They tried to make a new communication by bacteria sending bubbles to each other. Here, I'm here, do this, do that. And they tried to have bacteria that counts. 1, 2, 3, 4, 5. So if a bacteria has to cycle doing different tasks, like in the industry, right? It will know where it is, in what step it is. And we also took a long time to think about safety. How can you make a safer bacteria that you can actually use in nature, in real life? How can you quantify how safe it is and how ethically treat this? And all this is well documented. There are nice, beautiful wikis. I won't go into detail, but I think that all these projects, as much as many other projects, are really inspiring. The last project was to fight tuberculosis. There are 2 people from the team here. Raise your hands. These are world champions of synthetic biology. Right? One of them is an engineer. Two of them. Okay, one is a computer science, and another one is a biologist. And this is what makes the strengths of this approach.
And one's a designer.
Sorry? Sorry, a designer? Would I call you a biologist? Oh my god. No, a designer. Bio designer. And so this is just to cover up what we're doing in synthetic biology here. So I mentioned the item. But if we want to go beyond this very small community of 10-15 students that we can host here and have other people involved in synthetic biology, and not necessarily students in faculty of medicine or in university of faculty, not students even. It can be designers, it can be anyone that is interested in this. How can they learn? How they can learn what synthetic biology is? And Helena, okay, she's back from the lab, right? So she started a fantastic project.
Rafael.
With her friend there, wake up mode, and which is a game. It's a game, it's open code, and it has to do with learning how to craft biological systems and solve amazed with different adventures there. I'll show you one or two pictures. They're beautiful and graphic design by Ariel Lindner that was part of the team. But that's not enough. The idea is that this game is an evolvable game, just like Artemis said. So everyone can take the code and create more adventures here. And what we hope is that at one point the adventures will not be only about things that we know how to solve, but challenges that we don't know how to solve, but players will craft the right genetic network behind that will actually solve the problem. And learning by gaming, learning by playing is important, but there is some basic science to know. And again, rather than do this in a small classroom here, that we're now recording is a MOOC, so a massive open online course for non-synthetic biologists, non-students, everyone that wants to jump in and learn about this. And this is a great guy called Jake who's supposed to be the lab that is doing this. And last but not least, and I'll show you quickly the things. So just a bit about the game, just to inspire you. Go and look at it on the web. This is Jake. I won't show the video. Go and find out by yourselves. And the last project I want to tell you about, and the leader of this project is also here in the room. Ian, raise your hand. Everyone should see who you are. Okay. So, IGEM, for those who sort of understood, that's a very big, heavy organization, supposedly open, organized by a team that used to be affiliated to MIT. There's 200-something teams there, different tracks. There's a new track for art and design in this competition. It costs something like $30,000 to $50,000 to actually do this adventure. Okay? For DIYers, for designers, for anyone that is really passionate in this but does not have a lab that will host him, there are many better things to do with 30,000 euros than to spend them on such a competition. How can we put these people together? How can we work together on ideas? How we can break the wall of the university, basically saying that what matters when you do research, right, is the idea, the design, the question, and the project. And if it happens that a certain lab has the right tools, the right know-how, how to make this project work, let's couple these people together. Let's make this kind of pro-am collaboration actually working. So this is a project called SynBio for All. I invite you all to see this. This kind of perspective workshop enters directly at the heart of this project, right? Putting people that will never think they will find themselves in the Faculty of Medicine actually working together. The SynBio for All project takes it one further step. We ask people there to come up with their ideas, make their projects, build it inside the community that will peer review the project, and at least our lab here will adopt one project, will bring people over, no matter where they are, come to join us in the summer and make this project work. And hopefully this will be a new way of thinking about how to do science, citizen science, and joining forces with great brains, great ideas, and great technology. And I'll stop here. Thank you very much.